Hello everyone! Today I wanted to talk about optogenetics. This is a fascinating technique of neuroscience that has already helped grow the field of neuroscience so much, and I hope will continue to do so and be a great tool for future research. The learning objectives for this video are to understand what optogenetics is and how it works, and to understand how optogenetics has already been used in research so far. So, to start with, what is optogenetics? In 2005, a technique was created that makes use of genetically coded light-driven ion channels in pumps, and these ion channels can either excite or inhibit neurons. So, we can use these light-sensitive proteins in neurons to monitor and control their activity very precisely. So, if you take a look at this picture up here, you can see that in all the way on the left here that these neurons are being electrically stimulated. So when neurons are electrically stimulated, all of the neurons in this area are going to activate. And this has its drawbacks because it's not very precise and it could even cause damage to the brain when electrodes are inserted. So moving on to this middle part here, you'll see that with just the light alone that none of the neurons are going to activate. But then all the way on the right here, you can see that neurons have been individually selected and given this light-sensitive protein that will activate to the light. So we can just put it in this one, and this one, making it much more precise to really study what's going on and really get detailed with what we want to look at. And as I mentioned before, we can either, depending on the protein that's inserted into these neurons, you can have different lights. So for example, let's say that the blue light is going to excite the neurons, and then let's say we also had a red light and the proteins were coded for a red light, and that red light could be inhibitory to different neurons. So how does all of this work? Basically, scientists use a process of genetic engineering to change information in the genetic codes, and that code allows neurons to make special proteins. The special proteins it makes are called opsins, and the opsins are what respond to the light. So opsins can be put into specific types of neurons or into specific locations of the brain. So we can really zoom in here and look at a really small level of just a cluster of neurons, or we can zoom out and look at larger areas of the brain. Optogenetics is instrumental in finding links between brain and behavior. Optogenetics has been used to understand how cell types, circuits, and systems operate in normal and pathological states. For example, we have learned that different cortical interneurons have distinct behavioral roles. It has also been used to study cell communication, both with individual neuron clusters and more broad interactions between larger brain regions. And there was actually a study done in mice that used optogenetics as a way to see what happens to the brain after we have a stroke, and how does the brain function after the stroke. There have been a multitude of studies done in animal models. I'm only going to talk about a couple of them. So there was a study done that showed an anti-anxiety response when cells were activated in the ventral hippocampus, which is pretty cool if you think about it. And then in this picture down here, we have a picture of a mouse brain, and you can see this area right here, all this pink area that's lit up. So those are neurons that have been given a, they basically have opsin, and you can see them lit up, meaning that the light is on, and it shows the inhibitory circuit between the thalamus and the prefrontal cortex. This is an important route to study because there are cognitive disorders such as schizophrenia that use this circuit. So this helps us conclude that optogenetics could be valuable in treating or at least understanding mental disorders at a greater depth. So, in conclusion, optogenetics is a game changer for neuroscience. There's so much that we have learned over the past almost 20 years and will learn from the advancement of this field. Thank you guys so much for your time.